so much for being here. It's a pleasure. I'm very excited to talk about our research. So my name is Alexei Koulakov. I'm a theoretical physicist and applied mathematician by training. And my colleagues and I will work at Cosmic Harbor trying to figure out how the brain works. Today I will talk about a subject which I thought was relevant, which is sense of smell. And I have a video right here. I should say that this video is not up to scale, so nothing is really that big. And it also was made by a real human being sitting there. So the molecules in the environment, they float into our nose, and then they land in our olfactory epithelium, where they interact with a specialized set of neurons, which are colored here, you can see. And the reason why they are colored here by different colors is because they produce a specialized protein called odorant receptor. This protein will transduce the information. It will interact with those molecules in the environment, and it will be activated, and it will produce electrical pulses in those neurons. And you can see that there are many different colors. In our retina, we have only 3 color sensors, right? 3 cones, most of us. But in the olfactory system, there are 350 different types of olfactory receptors that have specific interactions with different molecules. So olfactory system is enormously complex. And then the cells, each cell makes a decision which protein or which odorant receptor they will produce, and therefore it will be color by color. And all the cells that have the same odorant receptor, they will converge on this bubble in the brain, in the olfactory ball, it's called. So this bubble is called glomerulus. So the surface of this brain region, which is responsible for odorants, is covered by those bubbles. So they look like pixels that they cover the surface of this brain region. And by the pattern of activation of those spherical structures, we can figure out what the smell is in the environment. So that's the basic idea. So these neurons, I have to be careful about pressing the right button. These neurons, so there are 350 colors, there are 350 genes in our genome that interact, that are responsible for recognizing smells. And the particular pattern of activation will tell us what is the smell which is lingering in the environment. So roughly speaking, if our vision is based on the retina, on the responses of neurons in the retina, which are, there are about 1 million, we have about 1 megapixel in the retina, and there are 3 colors that we can recognize, like red, green, and blue. So olfaction is, roughly speaking, there are 4 pixels and 350 colors. So it's very difficult for us to comprehend what this enormous color space is doing. And so these bubbles that are activated in odor-independent fashion, they will then send this information, will derive some other neurons, and they will send this information to olfactory cortex, and then the cortex will figure out what is the actual stimulus present in the environment. Like this flower, this beautiful flower in this particular case. So just to describe you why this problem is so complex. So this is comparison of the olfactory genes, olfactory genomes in different animals. So this is our eye. So our eye has 3 color tones, which are roughly red, green, and blue. So the Drosophila, the fruit fly, has 61 of odorant receptors. So they're colored here by those, schematically by colors. Our genome, our human genome has 350 different types of odorant receptors. Those are only the ones that are working. We also have an enormous number of receptors that are not working, and they're shown here by empty circles. So our genome is basically sprinkled with the remnants of our former olfactory glory. At some point we were very good at recognizing smells, but not anymore. Not as good as dogs and mice. So mice have about 700 colors. Dogs have about 700 different olfactory odorant receptors, and mice have about 1,100 odorant receptors. And I think record belongs to African elephant, who has more than 2,000. And this just underscores why olfaction is such a misunderstood or understudied subject is because the number of colors, the number of molecular complexity of the sense, the number of genes that we have to study is so enormous. Right? So the interesting thing is that the ironic thing that is to explain olfaction to you, I have to use vision, right, and auditory sense. But like if I'm in the same position as, for example, if mice had to explain vision to another mouse, because mice are very olfactory animals, right? So imagine a mouse explaining, like describing Mona Lisa, the painting, to another mouse. How would it do it? Her smell is so mysterious. It's masterpiece. So, but it's important to understand the sense, however. It's important both fundamentally, because it's just part of us, that we have no understanding how it works. It's also important because it's involved in many conditions such as Alzheimer's, Parkinson's, autism, so there are persistent defects in all those neurodevelopmental and neurodegenerative conditions that are also somewhat poorly understood. There's also a lot of imprinting, like sexual imprinting, including in the human population, which is dependent on olfaction. And finally, we at the lab we work to build biosensors that would allow to recognize various conditions such as cancer or Parkinson's just based on smells using animal olfactory systems. So I will talk about it later a little bit. So this is how we try to approach this problem. So we're trying to build something which is called the smelliverse, which is the universe of smells. So this is the space in which we think the smells belong to in our internal perceptual space. Like for example, the smell of lemon would probably be very close to the smell of lime in that space, but would be further away from the smell of apricot or pineapple, right? So this is our internal perceptual space. And we're trying to understand what the space is, and today I will talk about two dimensions in that space. And space is replicated here by this object, which scientific name of it is a potato chip. It's a two-dimensional structure, and individual dots there that you can see are the individual smell molecules. Okay? So there are about 150 of those smell molecules which are sprinkled around the potato chip. And it turns out that we actually know a lot about human perception, how those molecules are perceived by humans. And most of that information comes from descriptions. Just there are a lot of people that are meticulously describing thousands of molecules by words, right? And so if I look at the vertical axis and I read the words, so these are the words that describe molecules on top of this potato chip, on top of this two-dimensional smelliverse. So the molecules, I don't know if you can read this, but the words that describe the molecules on top are sickening, putrid, rancid, sweaty, pungent, and so on. And the bottom molecules are described by nice words. Rose, fruity, citrus, and so on. So it's kind of obvious that this axis represents something that is called pleasantness, right? So it's overall how good this molecule is for you. Should you run towards it or away from it? And this has been known for a while, that this pleasantness is a very important dimension for humans and for animals too. It's just goes directly into our motor function. And the other dimension is more mysterious, and I'm going to explore it with you, right? So the words that describe molecules on the right are like this: woody, resinous, burnt, musty, almond, peanut butter, oily, warm. And the molecules which describe the smells on the left are chemical, gasoline, fruity, cool, etherish, sour, putrid, fruity. So what do you think this dimension means? Any ideas? So I'm a mathematician, I'm a semantic idiot somewhat. So I usually ask people what things mean, things semantic mean. And so I have a lot of responses. Any hypotheses? Huh? They're more what?
The right are more pleasant than some of the left.
Yeah, some of them are more pleasant, but like burnt and smoky, I don't know.
They're drier. They're drier.
Right.
Earthiness?
So there's a lot of hypotheses that I heard. Yeah, edibility, like probably something related to kitchen. A lot of things that are related to kitchen on the right and on the left. But the most interesting hypothesis that I heard was this. I hope it's come up. Auditory pitch. So there's this idea that one of the important dimensions is actually tied to our auditory perception. And so the horizontal dimension is related to frequency. And so this is one of the examples of synesthesia, when two different senses are merged for some unknown evolutionary reason. And so I want to conduct an experiment with you. So if you don't believe me that there is an auditory pitch in olfaction, let me ask you this question. So imagine the smell of oil, for example, olive oil, and the smell of vinegar. Which smell do you think is lower in associated auditory pitch? Oil, right? So I guess it's pretty convincing. 90% of people usually respond to oil, and 10% they just don't understand the question. They reject the question. So this idea that there is this remarkable merging between the auditory sense and olfaction is pretty old. It belongs to a London perfumer whose name was Septimus Peace. So he, about like mid-19th century in London, he created the so-called gamma of odors, and he placed smells on piano keys, and he then played some chords on the piano, and that's how he created perfumes. And he was actually, Septimus Peace was in the news recently because one of his perfumes was discovered at the bottom of the Caribbean Sea. So there was a shipwreck of, so the ship, which name was Mary Celestia, it drowned around, I think it's like 1864. It was smuggling weapons for the Civil War. It was en route to Wilmington, North Carolina, so it sunk near Bermuda, and then it was lifted recently, and they discovered 2 flasks of perfume. One of them was closed. So the other one was opened, the perfume went spoiled, and the one that was closed, they opened it, and then they gave it to a professional perfumer, and the professional perfumer reverse engineered the perfume. So now we can smell this perfume, which was designed using this weird strategy of like playing chords. And actually we have a sample here, so if you, at the end of this talk, if you want, you can try to sample it, see if you like it. So I want to talk a little bit about animals. So in humans, as I mentioned, there's a lot of data about how humans perceive smells, but they just rely on like descriptions provided by words. But in mice, we can actually we can look at the responses of neurons in the mouse brain, and we can build the smelliverse, or the space of smells, based on the responses of neurons. And what you can see here are the dots here, individual smells. So this is the representation of responses of neurons in 2 mice. And the representations of responses for the same smell are linked by those lines. And as you can see, the neural responses in 2 different mice to the same smell are pretty close. This basically means that, so there is a possibility that mice experience the same smell very similarly, right? So which brings us to the question of qualia. It's like the question whether you perceive red in the same way as I do. So I guess we are trying to scratch the surface of that question in the world of smells. And we can say that mice perhaps experience the same smell very similarly. Well, there is some differences between them, but they're very, very small, very little. So this is the mouse neural smelliverse, which basically lacks the subjectivity associated with language. But then we live in the AI era, so we can actually translate those messages the neural responses into words using large language models. So here you can see some examples of taking the thoughts of the mouse, those neural responses that I showed you before, and then we use large language models and some algorithms that I used in encryption and decryption of messages. So we use decryption algorithms to decrypt neural responses and to produce the descriptions of smells as if mice were able to talk. Okay? So these are the descriptions, which I think, I'm sorry for the font, I'm not sure, I wasn't sure what the resolution of the screen is, but I'll read them for you. And this is the descriptions provided by the human expert, or rather by the head of the human expert. So this smell, the mouse described it like this. The odor is pungent, sour, and slightly rotten. The scent is fruity, cheese-like, thick fat. I'm not sure if it makes sense. But the scent, this is a human description. The scent is sharp, pungent, and sour, reminiscent of vinegar. So basically the mouse got pungent and sour, but it added cheese. For some reason, mice talk about cheese a lot. I don't know, but it's real. It's absolutely real. I don't know why. And then so it actually got two descriptors out of. So the next one, the scent is fruity, this molecule, and sweet with a pronounced odor reminiscent of citrus, green, and banana. The human description, the scent is strong, sweet, and fruity with notes of banana, pear, and pineapple. So the mouse got banana and sweet and that's it, and fruity, right? So most of the descriptors we can actually predict on the basis of neural responses in the mouse. And I guess we're trying to use this technology to really analyze smells, analyze smells around us, like for example, smells emitted by humans, with the potential to recognize whether humans have certain health conditions. So this is just scratching the surface. And mice are very good in olfaction, which I mentioned, because they have a lot of olfactory genes. So the hope is that at some point in the future, we'll be able to build those like biosensors where we can either identify a human or say a human what is wrong with it. So thank you very much. And if you would like to smell the perfume that was created by this sound strategy, somebody will just come around and they will distribute the smell sticks. Thank you.